Hello, good morning students. Today we'll start with the process of digestion. Digestion is a very important process because it is the one which provides us with energy which is required for performing various actions. We consume food in the form of pulses, cereals, fruits, vegetables, oils. That food is converted into monosaccharides, amino acids, fatty acids, glycerols, and which are absorbed by our digestive system and then it is oxidized to produce the energy. So the digestive system, it includes mouth, which has three pairs of salivary glands, pharynx, esophagus, stomach, liver, gallbladder, pancreas, small intestine, large intestine, rectum, and anus. Mouth breaks up food particles, assist in producing the language. Salivary glands produce saliva, contains amylase for digestion of polysaccharides. Pharynx is involved in the swallowing of food. Esophagus transports the food to the stomach. The stomach churns the food. Pepsin digests the protein. HCl activates the enzyme and breaks up the food, kills the germs. Mucus protects the stomach wall. Liver, it breaks down and builds up many biological molecules, stores vitamin, iron, destroys all blood cells, destroys the poison. Bile aids in the digestion. Gallbladder stores the bile, which is released into the duodenum. Pancreas, it produces both hormones as well as the enzymes. The enzymes like trypsin, chymotrypsin, carboxypeptidases, amylases, lipases are responsible for the digestion of carbohydrate, proteins, and fats. The membrane, the epithelia of small intestine, produces various enzymes like peptidase, sucrase, amylase for the digestion of proteins, sugars, and polysaccharides. Large intestine reabsorbs the water and ions and forms the fecal matter. Rectum stores and expels the fecal matter. Anus is the opening for the elimination of the fecal matter. So we'll start the process of digestion. Digestion is a process of gradual breakdown of complex food into simpler, soluble food with the help of enzymes for absorption. For example, bread, rice, wheat, and milk are chemically too complex and cannot be absorbed from the alimentary canal. So first they are digested before the absorption. The digestion releases its constituent like glucose, amino acids, minerals, salts, fats, vitamins, which are readily absorbed. So for the digestion, first process is mechanical digestion. It is mastication. The teeth, they are digested, they are designed for chewing. Incisors provide a strong cutting action. The molars provide grinding action. Chewing is important for digestion of food, but it is especially important for most fruits, raw vegetables because they have indigestible cellulose membrane around their nutrient portion that must be broken down before the food can be digested. Chewing aids the digestion of food by mixing digestive enzymes. Digestive enzymes act only on the surfaces of food particles. Therefore, the rate of digestion is dependent upon the total surface area exposed to the digestive secretions. Grinding the food to very fine particulate constituency prevents excoriation of the gastrointestinal tract and increasing the ease with which the food is emptied from the stomach into the small intestine and then into succeeding segments of the gut. So see here the process of mastication. So with the help of various muscles, the jaws, they help in the mastication. We intake the food. With the help of teeth, the food is mixed with the saliva during the mastication process. Bolus is formed. That bolus is thrown into the oropharynx and into the esophagus. Now swallowing. Swallowing is the complicated mechanism principally because the pharynx serves for respiration and swallowing both. The pharynx is converted for only a few seconds at the time into the tract for swallowing of the food. Tongue helps in the mixing of saliva with the food. Saliva moistens and lubricates the food, which changes into semi-solid form called bolus. The bolus is then swallowed through esophagus into the stomach. The peristalsis movement of alimentary canal also helps in the swallowing. So here we can see the peristaltic movement. Now see here, from the mouth after mastication, the food it enters into the esophagus, from where it moves to the stomach. Now here in the pharynx, basically pharynx is of three types, that is nasopharynx, oropharynx and laryngopharynx. So food is bolus is thrown into the oropharynx with the help of the epiglottis. So in the human digestive system, the main sites for digestion are salivary glands, gastric glands in the stomach, then pancreas, secretory cells you can say, and secretory glands of small intestine. So in this figure we can see three pairs of these salivary glands, parotid glands near the ear, then below the tongue sublingual gland, and in the maxillary portion submaxillary gland it is present. Now coming to the complex food substances which are taken by animals and humans must be broken down into simple, soluble, diffusible substances before they can be absorbed. They include the following: lingual lipase, salivary amylase, lysozyme. There are two types of salivary glands. One is serous glands and second is mixed gland. Serous glands, these are producing the secretion which are rich in water, electrolytes and enzymes. A great example of serous gland is parotid gland. Mixed gland, these glands have both serous cells and mucous cells, and they include sublingual and submandibular glands. Their secretions is muciginous and high in viscosity. So they produces lingual lipases, salivary amylases or ptyalin, and lysozyme. The wall of stomach undergoes periodic movement as well as contraction, producing churning movement called peristalsis, which results in breaking the complex food into the simpler one. The bolus, after mixing the gastric juice, turns into fine soluble form known as chyme. The chemical digestion involves the breaking of covalent chemical bonds in organic molecule by digestive enzymes. Carbohydrates are broken down into monosaccharides. Proteins, they are broken down into amino acids. Fats, they are broken down into fatty acids and glycerol, all with the help of enzymes. Now coming to the gastric enzymes, the stomach, it plays a role in digestion, both mechanical sense. by mixing and crushing the food, and also by enzymatic sense by digesting it. The following are the enzymes produced by the stomach and respective functions. Pepsin, the main gastric enzyme produced by the stomach cells called chief cells. In inactive form known as pepsinogen, it is a zymogen. It is activated in the stomach with the help of HCl and converted into the active form known as pepsin. Pepsin breaks down the protein in the food into smaller particles such as peptide fragments and amino acid. Protein digestion therefore primarily starts in the stomach, unlike carbohydrates and lipids which starts their digestion in the mouth. However, a very trace of enzymes called kallikrein which catabolizes certain proteins found in saliva of the mouth. So digestion of protein, milk protein takes place in the stomach with the help of prorenin, HCl, and peptones and proteases they are formed. Gastric lipase is acidic lipase secreted by the gastric chief cells in the fundic mucosa in the stomach. It has a pH optimum of 3 to 6. Gastric lipase together with lingual lipase comprise two acidic lipases. These lipases, unlike alkaline lipases such as pancreatic lipases, do not require bile acid and collapse for optimal enzymatic activity. Acidic lipase make up 30% of lipid hydrolysis occurring during digestion of human adults, with gastric lipase contributing the most of two acidic lipases. In neonates, acidic lipase are very much important and they provide 50% of the activity. There are 4 types of cells in the stomach. Parietal cells producing the HCl and intrinsic factor for the absorption of B12. Gastric chief cells, they produces pepsinogen. Chief cells are mainly found in the body of stomach, which is middle and superior anatomic portion of the stomach. Mucous neck or pit cells produce mucin or bicarbonate to create neutral zone to protect the stomach lining from acid or irritants in the stomach chyme. G cells produce a hormone gastrin in response to distension of stomach mucosa or protein and stimulate parietal cell produce their secretion. G cells are located in the antrum of stomach, which is the most inferior region of the stomach. Now see here, these are different cells, that is the mucosa neck cells, parietal cells, chief cells, D cells and G cells. Pancreatic juice, composed of the secretion of both ductal or acinar cells, contains the following digestive enzymes: trypsinogen in inactive form, chymotrypsinogen in inactive form, carboxypeptidase again in inactive form, elastases. Basically they act on the proteins. Pancreatic lipase, which degrades triglycerides into two fatty acids and monoglycerides. Sterol esterases, phospholipases, nucleases like DNAase, RNAase, pancreatic amylases. A very important thing is humans lack celluloses to digest the carbohydrate cellulose, which is a beta-linked glucose polymer. So pancreatic juice, it contains the enzyme for the digestion of carbohydrates, proteins, fats, nucleic acids, all. Throughout the lining of small intestine, there are numerous brush border enzymes whose function is to further break down the chyme released from the stomach into absorbable particle. These enzymes are absorbed while peristalsis occurs. Some of these enzymes include erapsin, which converts peptones polypeptide into amino acids; maltase converts maltose into glucose; lactase converts lactose into glucose and galactose; sucrase converts sucrose into glucose and fructose. Exocrine cells in the mucosa of small intestine secretes mucus, peptidase, sucrase, maltase, lactase, lipase, and enterokinase. Endocrine cells secretes cholecystokinin and secretin. The most important factor for regulating secretion in the small intestine is the presence of chyme. It is largely a local reflex action in response to chemical and mechanical irritation from the chyme and in response to distension of the intestinal wall. Chyle is milky bodily fluid consisting of lymph and emulsified fat or free fatty acids. It is formed in the small intestine during the digestion of fatty food and is taken up by the lymph vessel, especially known as lacteals. The lipids in the chyle are colloidally suspended in chylomicrons. Thank you. www.google.com for providing us beautiful pics so that we can explain our topic well.